Hi, my name's Ari Li, and I'm a sophomore at Yale University studying applied math and economics. This summer, I interned in Dr. Li's lab at Mount Sinai, where I was a part of the NIH SenNet CUSP summer internship program. This summer, my research was about understanding senescence through human lung xenium and uPAR data. So, some background about my lab. The Li Lab is part of the Tissue Mapping Consortium, specifically focusing on the lung and colon areas. So, previously to joining this Tissue Mapping Consortium, the Li Lab previously focused on lung and blood vessel aging and has since found senescence to be an area of interest. Some background is that the PI of the lab, Dr. Ari Li, used to be a clinical pulmonologist, but transferred over to the area of academia, which she found to be more interesting. So, an overview of senescence is that it can be characterized by an irreversible cell cycle arrest, which can be caused by a series of epigenetic factors such as radiation, UV ray exposure, and oxidative damage. But it can also be caused by a variety of normal, healthy bodily functions, such as aging, in which telomeres can shorten over time when will at some point turn a cell senescent. So, as part of the Tissue Mapping Consortium, the Lee Lab has the task of developing maps of healthy human tissues and senescent cells. However, as part of this task, several problems became apparent. One of these is the relative rarity of senescent cells in human tissues, which makes them somewhat difficult to map. The controversy as to which senescent markers would be used to determine whether a cell was senescent or not senescent, and the relatively low expression of these senescent cell markers. And in addition, the absence of these reagents, which can be used for determining these senescent markers. So, as a result, the 5 by 5 project came to fruition. And as part of the 5 by 5 project, there are three main goals. One of which is to establish the sensitivity of specific single cell and spatial mapping techniques by comparing assay performance in the same tissue at various time points following senescence induction. Now, that's a mouthful, but what it means is to basically look at various assays and determine how useful they would be when directly approaching the problem of senescence. Goal number two is to establish assay specificity by comparing the detection of senescent cell markers in the same tissue before and after senescence induction. So this is used to determine specificity by using the same exact test before and after you turn certain cells into senescent cells. And the third main goal is to explore the possibility of heterogeneity in senescent cell markers and SASP between different tissues with aging and with senescent cell induction. And so what this means is that between certain tissues, there may be, different senescent cell markers or gene expression in these SASPs that could become a factor when determining which cells are senescent cells between if you're looking at, lung or colon cells. And so the Lee Lab is not the only lab as part of the tissue mapping consortium focusing on lung and colon. And so if you look at the following table, you have several labs at Mount Sinai working on this. And so you can see below where you see Sojin Kim and Alter Klausner, our lab was specifically assigned to work on xenium. So what is xenium? Xenium is a newly developed spatial imaging tool, which we decided would be a great tool to use when studying senescence for two main reasons. So Xenium is able to detect gene expression of hundreds of variant senescence markers that we could custom select. And in addition, Xenium can detect gene expression on the single cell level, which is incredibly important when studying senescence because of senescent cells' rarity and their ability to be of, various certain cell types. So, for example, if you were to look at Visium, which is another spatial imaging tool that can measure gene expression, you can see that it's very hard to tell, where certain cells start and where they end. And in addition, you can't really tell what cells are what kind of cells. Whereas if you look to the right and you look at Xenium, you can directly see where the cell membrane is and see what kind of cell it is and where the gene expression is in that cell, which is incredibly important because it allows us to better map senescence in these tissues. So the first step of running Xenium is to create a tissue microarray where me and the Li Lab helped run an H&E where we created samples and cored them out, where each core had a radius of 1.5 millimeters. And we had a total of 114 samples between the two Xenium runs and 383 genes that we were detecting specifically. And 100 of these genes were custom selected to be senescence markers for the purpose of studying senescence. And of these regions, we had them annotated by a pathologist who was able to break down the cores into 41 alveolar cores, 37 bronchial, and 42 vascular. And you might see that this doesn't exactly add up to 114, and that's for the reason that there was some overlap. Okay, so looking at the data. So Xenium is a relatively new, it's actually our lab's first running of Xenium. And so we were looking at a test control model, and we wanted to first see that Xenium was able to be run and that we had all the necessary, protocol and steps in place to be able to run Xenium properly. So we have our first run and our second run, and they look pretty homogeneous when looking at the UMAP chart. And UMAP is a tool that we use to see if there are any differences between the overall gene expressions of each sample. And if you look to the right, we have, specific cell types all in different areas of UMAP, which is really expected because this shows that, each cell is like expressing different groups of genes, which is a really good sign that our Xenium was working. So this to the left is our overall Xenium data, and to the right is a previous single cell analysis that we used to compare against the Xenium to see that each cell type was adequately represented. So in addition to checking that each cell type was adequately represented, we wanted to see that our cell specification system was working properly. So when we were running with the Xenium, there was a really large amount of cells, and it was impossible to be able to look through each cell and determine what cell is each kind. So we had to automate it with a process and to perform a sanity check to see if our process was accurate. We broke down each cell type and studied its gene expression with UMAP. And we were able to see that each cell type was pretty clustered and the specific cell types were expressing the right genes, which showed that the Xenium was categorizing each cell type by its correct cell type. So in the future, The Li lab intends to run Xenium with human lung samples from bronchoscopy biopsies and run Xenium human colon from colonoscopy samples, analyze each data set from the Xenium runnings from the prior two steps to map out these senescent cells and to compare the data sets between lung and colon samples to see if there are any specific biomarkers that you may see in the colon samples and specific biomarkers that you can see in the lung samples that, may differ, which may show some kind of correlation that we could further look into. And finally, we will compare the human and mouse xenium data. So our lab's next area of focus is uPAR. So what is uPAR? uPAR is a surface protein located on the cell membrane, which is used to regulate the degradation of the extracellular matrix. And so our lab found uPAR to be an interesting area of study because the gene encoding uPAR, plower, has been shown to be upregulated in senescent cells. And why does this make uPAR interesting? Well, because it allows uh, uPAR to be a target for senescent cell therapies or senolytics, which can directly target senescent cells and clear them out. And in addition, uPAR has also been found to not be expressed in many vital organs, which is a really good sign because it allows uPAR to be uPAR therapies. So senolytics that directly target uPAR as a surface protein to be relatively safer as they won't target too many outside cells. So the current idea with uPAR is to use CAR T cell therapy. So the rise of CAR T cell therapies has originally started as cancer immunotherapy, where CAR T cells were specifically targeted to attack cancer cells, which would decrease the amount of collateral damage that traditional chemotherapies would have. And so the thing about senescent cells is that senescent cells are relatively rare, and so we need to find very, very specific and targeted therapies to remove them, or otherwise there would be incredible amounts of collateral damage if we were to use some sort of chemotherapy approach. To first start off to see even if uPAR is a good target, we analyze our Xenium results. So from the results from our first running, one of the genes we targeted was plower, which encodes for the uPAR protein. And what we saw was that plower overlaps heavily with sen-mayo, which is a senescent cell statistic that is a good indicator for whether a cell will be senescent or not. And if you look at the two pictures, you'll see that there is a high amount of overlap between the two, which is a really good sign as to uPAR being a promising target for senescent cell therapies. And so in the future, what we hope to do is we hope to run immunohistochemistries to directly see if uPAR exists on these surface proteins before we run more assays and use more samples to verify the existence of uPAR. And so what this means is that previously we had looked through Xenium to see if the plower gene was expressed in these senescent cells, but now we need to see if the uPAR protein itself is actually on the cell membranes of these senescent cells, which is, a pretty big difference. And actually, our lab should be running our immunochemistry within the next week. So I am excited for those results. In closing, I would like to thank the NIH SenNet CUSP Summer Internship Program, as well as the CUSP managers that were able to make this possible, especially Elizabeth Martinez and Kay Mati, who really ran the program really well and kept everyone on track. In addition, I would also like to thank my lab mates for keeping me going throughout the research process and really guiding me through every single step of the way, explaining to me, every part of the experimental design process, all the way to, the micro adjustments for the xenium. And I'm really inspired by them and am really considering a graduate degree in biostatistics now that I see a way forward in academia. And I'm really grateful for this summer's experience with my research. Thank you.